day, everyone. This is Jano Clemenia, and I'm here at Biotech, Center for Studies in Biotechnology, Cebu Technological University, Barili Campus. And today, I'm going to demonstrate the technology of tissue culture. Tissue culture is the cultivation of plants in vitro, which is the Latin term for within glass, in artificial medium under aseptic and controlled condition. Among the basic techniques or kinds of tissue culture were the shoot tip culture, embryo culture, callus culture, cell culture, and the protoplast fusion. It is specially developed and made possible based on the two fundamental abilities of plants, the totipotency and dedifferentiation. Totipotency is the potential ability of plants to stimulate a cell to regenerate again into an entire plant, while dedifferentiation is the capacity of mature cells to return to meristematic condition. First initiated by the father of tissue culture, Gottlieb Haberland, tissue culture had been an important tool in all sorts of plant and even animal breeding. The importance of tissue culture is that we can mass-produce plants in a large-scale basis. It is virus-free and clean and allows the rapid release of improved clones or genotypes. The basic steps in tissue culture always begins in the preparation and pre-treatment of planting materials. It will undergo surface sterilization before proceeding to inoculation and then incubation. And when shoot initiation will occur, further multiplication of plantlets will be maintained to mass produce new plants. One factor that should be considered is the culture medium preparation. Listed in the table are the basic ingredients of the well-known Murashige and Skog medium. Specifically, the culture medium will be composed of the macronutrients, micronutrients, vitamins, iron EDTA as chelating agent, sugar as carbon source, agar for solidification of medium for the upright growth of plants, and other undefined components such as coconut water. These ingredients will be weighed in an analytical balance and will be sterilized in the autoclave at 15 psi for 20 minutes. And for this part, I will now show you how to do tissue culture for specific crops. Step or stage 0 in doing tissue culture is the pre-treatment and preparation of planting materials. I have here a plant sample. This is an abaca, and we're going to conduct a simple sterilization procedure before we're going to proceed in the inoculation process. Sterilization of our plant sample, we're going to use liquid detergent, commercial bleach, and ethanol. After the sterilization process, sample inside the laminar flow for further sterilization. And now we can proceed to the inoculation process. Beside me is the laminar flow hood, where we are going to conduct the inoculation of our plant samples. After the inoculation process, we can now place our plant samples in the incubation room, where they will be maintained and monitored for growth and later be mass-produced for new plants. And that is how we're going to do tissue culture. Thank you so much for listening, and I hope that you learned something today. See you again next time.